Welcome to our lecture on the biochemistry of metabolic processes. Today, we are diving into the metabolic fates in liver cells. Imagine your body as a massive, bustling city. For this city to survive, it needs a highly intelligent central distribution warehouse to manage its fuel and resources. That is exactly what your liver does. It is the ultimate metabolic multitasker, constantly shifting its complex biochemical machinery to meet your body's ever-changing systemic needs. Let us explore how this incredible organ keeps us alive through feasts and famines. Now that we understand the liver's overall role, let us look at how it functions as a central warehouse. When you eat a large meal, your digestive system absorbs various nutrients. The liver rapidly sorts these packages. It stores the excess energy so it doesn't overwhelm your bloodstream and cause damage. Conversely, when you are starving or fasting, the liver breaks into its emergency reserves. It even manufactures brand new fuel from scratch to keep your brain alive. Without this central metabolic traffic controller, our blood sugar would wildly fluctuate, and our vital organs would quickly starve between meals. Building on this warehouse analogy, let us examine the specific enzymatic pathways for different nutrients inside the hepatocyte, the primary liver cell. For glucose, the liver can convert it into glycogen for safe storage, run it through glycolysis for immediate energy, or convert the excess into fatty acids for long-term storage. For amino acids derived from dietary protein, the liver synthesizes vital structural proteins in the blood. It also converts their carbon skeletons into glucose through a process called gluconeogenesis and safely packages their toxic nitrogen waste into urea, preventing ammonia buildup in your body. Having seen how carbohydrates and proteins are handled, we must now look at lipids and how all these processes are controlled. For fatty acids, the liver oxidizes them via beta oxidation to generate massive amounts of energy. During prolonged periods of fasting, it converts them into ketone bodies to fuel the brain. The direction and magnitude of all these pathways are tightly regulated by hormones like insulin and glucagon. These hormones alter the phosphorylation state of key regulatory enzymes, effectively switching the liver between a fed state and a fasting state to maintain perfect systemic balance. Understanding these pathways is not just academic. It brings us to their critical clinical relevance. The liver's ability to seamlessly switch between storing and producing energy is what allows humans to sleep through the night without dying of severe hypoglycemia. However, when this delicate regulation breaks down, metabolic diseases occur. In diabetes mellitus, we see dangerous spikes in blood glucose and the toxic acidic accumulation of ketone bodies. Furthermore, conditions like fatty liver disease arise when the liver's capacity to export synthesized lipids is overwhelmed by excessive caloric intake, causing fat to accumulate and destroy liver tissue. To truly appreciate the liver's work during fasting, let us look at the mathematical reality of building new fuel. This chemical equation represents the overall stoichiometry for gluconeogenesis, which is the synthesis of one molecule of glucose from two molecules of pyruvate. The formula shows pyruvate, a 3-carbon precursor, being converted into a 6-carbon glucose molecule. Notice the ATP and GTP. These are the high-energy currency molecules of the cell. NADH provides the necessary reducing power. In plain English, this formula tells us that building a single glucose molecule is highly expensive, costing the liver a total of 6 high-energy phosphate bonds. If cellular ATP drops, this forward reaction will halt. Now that we have the stoichiometric formula, let us apply it to a real-world clinical scenario. A patient has been fasting for 18 hours, and their liver needs to synthesize exactly 3 moles of glucose to maintain adequate blood sugar for the brain. We need to calculate the total high-energy phosphate bonds required. From our equation, synthesizing 1 mole of glucose requires 4 moles of ATP and 2 moles of GTP, giving us a baseline cost of 6 moles of high-energy bonds per mole of glucose. Multiplying the 3 moles of glucose needed by the 6 bonds required per mole, we arrive at a final answer of 18 moles of high-energy phosphate bonds. This massive requirement explains why the liver must actively break down stored fats during a fast. This massive energy requirement of 18 moles of ATP brings us to an important question. How does the liver pay for this? The liver cannot rely on its own small stores of ATP during a fast. Instead, it must actively break down stored fats to generate the continuous stream of ATP needed to keep the brain supplied with newly synthesized glucose. To pay for the high-energy phosphate bonds calculated in our previous example, the liver oxidizes palmitate, which is a standard 16-carbon saturated fatty acid. The complete beta oxidation and subsequent cellular respiration of 1 mole of palmitate yields exactly 106 moles of ATP. This is a prime example of metabolic coupling. To understand exactly how much fat is needed, we use the metabolic coupling equation. This formula calculates the ratio of the total energy required by the liver to the energy provided by a single unit of lipid fuel. In this equation, N represents the number of moles of palmitate required to fuel the process. E sub T is the total energy demand, which is the ATP cost we calculated previously. The variable Y represents the energy yield, which is the ATP produced per mole of palmitate oxidized. This formula tells us in plain English what fraction of a fatty acid molecule is needed to pay the energetic debt. If the yield Y were lower, the liver would have to burn a much larger amount of fat to survive. Let us put this metabolic coupling formula into practice using our previous numbers. We know our total energy demand is 18 moles of ATP to synthesize the required glucose. We also know the energy yield is 106 moles of ATP per mole of palmitate. By substituting these known values into our equation, we divide 18 by 106. We find that approximately 0.17 moles of palmitate must be oxidized to meet the energy demand. This beautifully illustrates the incredible energy density of lipids compared to carbohydrates. The liver only needs to burn a tiny fraction of a mole of fat to generate enough energy to synthesize 3 entire moles of glucose. Now that we have calculated the energy costs, let us visualize how all these pathways connect inside the cell. On screen is a schematic of a hepatocyte. On the left, you see the bloodstream delivering 3 main packages: glucose, amino acids, and fatty acids. Follow the arrows inside the cell. Glucose is routed upwards to glycogen for storage or downwards to pyruvate. Amino acids are split. Their toxic nitrogen goes to the urea cycle, while their carbon skeletons become pyruvate for gluconeogenesis. Finally, at the bottom, fatty acids undergo beta oxidation to become acetyl-CoA, which either enters the TCA cycle for massive ATP production or is converted into ketone bodies that are exported to fuel the brain. This diagram perfectly captures the liver's multitasking nature. We have covered a tremendous amount of biochemistry today, so let us summarize our key takeaways. First, the liver acts as your body's central metabolic warehouse, constantly shifting pathways to regulate blood chemistry and prevent starvation. Second, we learned that manufacturing new fuel through gluconeogenesis is highly energy intensive, costing 6 high-energy phosphate bonds per glucose molecule. Third, we proved mathematically that lipids provide incredible energy density, allowing the liver to burn just a fraction of fat to sustain massive glucose production. Finally, all these interconnected pathways are tightly regulated by hormones to ensure your survival. You now have a solid understanding of the molecular basis of human nutrition and fasting. Great job!